I'd like to thank the organisers for the opportunity of presenting to you the data that we've obtained from our initial study of the impairment of neuronal architecture in the Snord116del mouse model for Prader-Willi syndrome. Loss of one or more genes from the Prader-Willi locus on chromosome 15 results in the metabolic, endocrine and cognitive impairments associated with Prader-Willi syndrome. While the metabolic and endocrine impairments have been fairly well studied, that cannot be said for the cognitive impairments. We know that individuals with Prader-Willi syndrome present with reduced IQ score and underattainments in numeracy, literacy and comprehension. Similarly, awareness of social norms is underdeveloped, and interestingly, some specific skills are more severely impaired than would be predicted by their IQ score. For parents and carers of children with Prader-Willi syndrome, this is clearly an issue. And as one advocate reviewer said, while hyperphagia is a constant worry and stressor, the cognitive deficits being explored in this study are just as debilitating in some respects. Cognitive function arises from three basic processes: the ability of nerve cells or neurons to receive information via the incoming dendrites, and then processing that information through the soma and axon hillock, and then transmitting that information to neighboring neurons via the axon and the terminal dendrites. Studying the function of neurons in this context is difficult in children, but can be achieved in rodent models of Prader-Willi syndrome. And mice with single or multiple deletions of genes within the syntenic chromosome 7 PWS region have been used to study both endocrine and metabolic dysfunction. Recently, a paper was published describing cognitive deficits in mice bearing a specific deletion of the Snord116 cluster in the PWS locus. And loss of Snord116 expression leads to impairments of novel object recognition, location memory, and tone cue fear conditioning. We wanted to study, therefore, the hypothesis that these cognitive impairments in this model may be due to either defective neuronal architecture and or defects in neurogenesis, that is the process of developing new neurons. In the first arm of our study, we took male mice bearing a deletion in Snord116 on the paternal allele and compared them to wild-type males. And at 10 days of age, the mice were killed and the brains collected. After being frozen, 150 micrometer cryostat sections were obtained, and these were impregnated for 2 weeks with Golgi stain. This produces neurons that have a beautiful black structure that is exquisitely detailed. We then performed Scholl analysis, and we did that on cortical pyramidal neurons. So those are the neurons whose cell bodies have this pyramid-like structure. And we analyzed cells in layers 2, 3, and 5 of the cortex. So that's the region of the brain responsible for motor function. And essentially, Scholl analysis works by placing a series of concentric rings around the cell body. And these rings differ by a 10 micrometer radius. So for the Scholl analysis, 30 neurons minimum were analyzed per mouse. And the data obtained from Scholl analysis is as follows. So we separated the apical dendrites, that's the dendrites that are conveying information from the cell to neighboring cells. And we showed that while there was a reduction in the number of primary dendrites, the overall dendritic number, branch points, and dendritic length were not significantly affected. And that is reflected in the Scholl arborization profile, where there is little difference between the wild type and the Snord116del males. In contrast, in the basal dendrites, so that's the dendrites that are receiving information from neighboring cells, there was a 15% reduction in dendrite number, a 20% reduction in the number of branch points, and a 22% reduction in dendritic length. And this is reflected in the Scholl arborization profile by a significant reduction in the number of dendrites from 30 micrometers to 90 micrometers from the soma. So loss of Snord116 compromises the architecture of the basal dendrites. In the second arm of our study, we maintained the female mice derived from the matings in the first half of the study, and we maintained them until they were 15 weeks of age. These were then killed by perfusion fixation, and after cryopreservation, the brains were frozen and 30-micron sections obtained. We analysed 6 sections per hippocampus for each of the different proteins that we were going to analyse. And we're particularly interested in this dark line of cells that's presented here in the dentate gyrus. We quantified cell proliferation using Ki67 as a marker of this process, and we stained for DCX as a marker of the number of immature neurons. We then subdivided the DCX positive cells using the method of Plump et al. In order to quantify the neuronal morphology. So this method quantifies the extent and arborization of the various dendrites that are protruding from the cell. Our data showed that loss of Snord116 has no effect on the process of cell proliferation, so Ki67 positive cells are unaffected. In contrast, loss of Snord116 results in an 11% increase in the number of immature neurons, that's DCX positive neurons, in the dentate gyrus. Although the number of animals we were using was low, and possibly is the reason why we didn't obtain statistical significance, our data seem to indicate that there is a shift in the morphology of the neurons from those with very little processes or arborization towards those where there is a more highly developed process and arborization. So in summary, our study indicates that loss of Snord116 results in a reduction in the arborization of the basal dendrites in the cortical pyramidal neurons of postnatal male mice. In contrast, loss of Snord116 results in an increase in the number of DCX positive immature neurons in the DG of adult females. And it's possible that this may be accompanied by an acceleration of the maturation of the dendritic tree structure. The take-home message is that loss of Snord116 has opposite effects in the cortex and hippocampus, and that these effects may also be influenced by the stage of development. So what are the implications? Well, loss of Snord116 may limit the ability of pyramidal neurons in the cortex to receive incoming information and thereby reduce cognitive performance. But this raises the question as to whether this deficit is seen in females, whether it's maintained into adulthood, whether it's intrinsic to the pyramidal neurons or dependent upon the developmental context in which these neurons are maturing, whether it's reflected in the electrophysiological function, and whether it's recoverable by manipulating those transcription factors that are known to control the maturation of these neurons. In terms of the hippocampus, loss of Snord116 may advance hippocampal neurogenesis in the adult and thereby either enhance certain behaviours or disrupt the normal processing and cell alignment within the DG. Again, this has implications for future direction, so it raises the question as to whether the enhancement that we have seen is observable in males, whether it's observable before puberty, whether it's reflected in the number of cells maturing to express new N, whether it's reflected in learning processes that are dependent upon adult hippocampal neurogenesis, and again, whether this process can be normalized by manipulating transcription factor signaling. We have to say we're very excited that the Foundation have agreed to fund our continuing study on this work, and we look forward to the data that we're able to achieve in this project. I'd like to thank in particular Janine and Blanca for their hard work in performing the Scholl analysis, and Miriam and Martina for their immunohistochemical analysis of hippocampal neurogenesis. I'd like to thank the Prader-Willi Syndrome Association here in the UK and the Foundation for Prader-Willi Research and Swansea University's Medical School Capstone Project for funding different parts of this study. Well, thank you for listening.